Hello, everyone. Welcome to the MIT IoT seminar series. We will be starting now. It is my great pleasure today to introduce our speaker, Dr. Jules Jaffe. Dr. Jaffe is a research oceanographer emeritus at the Scripps Institution of Oceanography at UC San Diego. He has a long and highly decorated career as an oceanographer, research oceanographer, since he received his PhD in biophysics from Berkeley. His work has encompassed over 20 systems for underwater optical imaging, acoustic imaging, tracking, and miniature passive robots. He has done amazing research. His 2002 article on laser sheet microscopy was cited by Nature as a milestone in microscopy. Dr. Jaffe has also keen interest in communicating his work to the public. His research has been featured in the American Museum of Natural History, the London Science Museum, including both traveling and permanent collections. Dr. Jaffe, thank you very much for joining us today. I'm very excited about your talk. Thank you for joining us virtually at MIT to give it. The screen is yours. Please take it away.
Okay, so you guys can see my PowerPoint screen. So thank you very much for inviting me. I know Fidel speaks fluent French, so I guess I just want to make one joke that my being here was a Fidel accompli. Oh, that was bad. That was bad. That was bad. Okay, let me get away with it. All right, so the name of the talk is Ocean Exploration and the Important Role of Technology Advancement. Okay, moving right along here, if I can, somehow that doesn't ever work. Okay, so you guys know all this, but I just want to provide some perspective. Um, it's not something that you wouldn't know, but when you think about, I started sort of in electron microscopy and medical imaging, and that's a whole nother story that we don't have enough time to talk about. But when you stick somebody inside of a CAT scan or a PET scan or an MRI, you're really from the outside looking in. And what you can visualize is the entire three-dimensional structure of that person or thing, or, I mean, a lot of us have experience with non-destructive tests. But when you're talking about doing oceanography, you're sort of on the other flip side of it. I mean, you can argue which was the inside out or the outside in, but what I wanted to portray was this idea that we all know that when we study the ocean, we're looking out as a participant on the inside. And that makes life more complicated but perhaps more interesting as well. So I don't think it makes sense to go through all the stuff I've done over my 30 plus year career because let me just say, some of them worked great, some of them almost worked, and some of them didn't work at all. But I think it's important to have a perspective that if you're not failing sometime, you're perhaps not reaching far enough. And I don't think we should be afraid of failing because if everything we do succeeds, then well, we could give ourselves a pat on the back, but maybe we could have done more. So on the left are the line scan lasers, underwater imaging systems. I started back in '85 with Ballard and Argo as a resident physicist, and moved here to Scripps in '88, and have been here ever since. And I have a low inertial mass for starting new things, and I'm continually fascinated with the advancements of hardware, technology, and software, and the increasing need for ocean exploration. So on the left, we'll show a bunch of imaging systems from macroscopic to microscopic. I even proved that you could get scatter from 50 nanometer particles and size them. On the right, we have a number of passive and active systems that I've developed through the years. And we sort of got interested in robotics because we wanted to be in the same frame as the organisms that we were studying. Because if you're sitting on a ship, stuff is going to be passing you. So the idea was to be quasi-Lagrangian. And I find that perspective to be the right place to be. Okay, so moving along. This is a pretty famous diagram. On the lower x-axis, we have we have, metric in terms of scale, and on the vertical axis we have time. This really started with Henry Snommel, who is a very famous oceanographer from Woods Hole. And I'm sorry I didn't get a chance to meet him. The other one was our own Walter Monk. And I sort of decided the fun place to work would be things that I could view around a week, and maybe around, millimeters to meters. And so I sort of like to hang out in this area. I okay. Okay. So I am not an expert on global warming and geophysics, but I thought it'd be fun to just show a simple little slide where we could make ourselves feel good that we're studying something that's important. And what's really important in the world today is the carbon dioxide and the greenhouse effect. And the ocean actually sequesters a lot of carbon dioxide. You can go online, and people who know more about that than I, which isn't very hard, can tell you what the facts are. And so here I'm showing the carbon dioxide being, photosynthetically activated by large and small meaning, phytoplankton, meaning sun-loving plankton, even down to the bacteria. Sometimes these guys get eaten by things that you'll be seeing in a few minutes. And sometimes it actually goes down and it's deposited into the depths of the ocean. So this whole scale here is vitally important for our Earth ecosystem. And so we think that it's really important to understand it. And the wonderful thing for us is that I believe oceanography is still a discovery-based adventure, because there's a lot of things that we haven't seen, and that the role that technology can play in providing that information to people like scientists that I work with, I think is an adventure that I've enjoyed my whole career. So that's sort of the wrap on that. Who cares about what? Well, the rate of harmful algal blooms are increasing. You may be aware these areas become anoxic, and we have dead fish. We're working with the Swiss, who are terrified that their lakes will be polluted with water-soluble toxins called microcystins. We are worried about showing oxygen, because the guys we study actually have to go deep enough so that they're not seen during the daytime. So they make a commute at dusk and they go back down at dawn. Many of us know the stories of coral reef degradation and how sad that is, mainly due to temperature, but we're also worried about acidification. And in our interest in attempting to preserve the wonderful things on the planet, we've set aside marine protected areas, and we were trying to figure out if they're really working, which I'm not convinced we know the answer to. There's a lot of fundamental questions in that as well. Okay, so this is the Scripps Plankton system, and the chief designer of that is Paul Roberts here on the left, who spent 20 years with me as an undergraduate, master's, graduate student, and then 10 years in my lab as a senior engineer. And on the right is Francesco Pomati from EWOG, if you're familiar with ETH, one of the premier intellectual universities in the world. EWOG is a freshwater adjunct to ETH. And what you're seeing here is this system that we've developed that they're holding in their hand now, but I don't really want to spend the time to go through each one of these things. But we're not really getting rich doing this. I want to make that point. If you want to start a company, go figure out how to do something that has more commercial possibilities. It's really a labor of love, and we more or less do this for cost, but we have derived a lot of pleasure from seeing the proliferation of this instrument and its impact in scientific studies. So that's Paul and Francesco sitting in Zurich in a lake. Okay, so the Scripps Plankton camera, if your engineers are also divers as well as designers, then they can go stick these things on something like the Scripps Pier. Here's Paul. These are the pier pilings. They're concrete, actually, that our Scripps Institution of Oceanography has underneath our pier. And I think it's kind of a fun picture of Paul. What you're seeing here in the bottom is we have a two-part. So I decided to have a free flow imaging system. which means stuff can sort of drift through and not be pumped through a chamber or when I sort of like this idea of seeing things moderately undisturbed, although, we do get some turbulence around these housings. And via that, we've had some remarkable images, which I'll show you in a moment. So we have two systems. This one on the left is a 2.5 centimeter by 2 centimeter field of view. And the one on the right is a 2.25 millimeter by 2.25 millimeter field of view. And this is really 10-year-old technology. So by the time you think of something, write a proposal, get it built, stick it in the water, get results, you're looking at least 3 to 5 years. And we were delighted to have this come to the fruition. These two organisms that I'm showing you, the one on the right, it's totally fascinating to me because what you're seeing is a organism which has an exoskeleton, right? So we invertebrates have a interior skeleton, and when we grow, we just grow from the inside out, and our bones get bigger. But if you're a lobster or some organism that has a crustaceous exoskeleton, you have to shed that. I was actually out on Cape Cod a couple of weeks ago, and there are all these skeletons from limulus, the horseshoe crabs. And I was like, oh, there's all these dead horseshoes on the beach. And one of the gentlemen I was talking to was, no, they're not dead. They just shed their exoskeleton. So this poor little copepod, which is about 1 millimeter in size, is squeezing itself out of its shell to be able to grow into a bigger organism. So you thought you had it hard. Be thankful that you're a vertebrate because this has got to be a hard thing to do. The one on the lower left couldn't be identified for a long time. This is probably around a couple of 100 microns. It's a multicellular precursor for sand dollars. So if you ever walk on the beach and you see those beautiful sand dollars, well, we all started small, as you know, and I thought this was sort of a fun picture. These are actual little nutrients. So I just want to, let's look at some pictures. I mean, my whole thing is imaging, and I thought it'd be fun to just show a bunch of pictures from these two cameras because I think people really respond to visual imaging. So this is off of our pier, and I want to make the point that we have a real-time stream, and this is not like Jules sat at home and picked out the good images and juxtaposed them all. Our system automatically segments and sizes the images that are coming in real time and displays them in that way. So what you're seeing here is a bloom of these helical diatoms and a few of these copepods, the guy that was getting out of his exoskeleton. And so it's really fun for me. And when I started doing this, the microscopists, I know people that collect these things, wow, we never see this in the field because when we collect the organisms, we break them up. So it's a sort of fascinating idea to me that there's kind of this fundamental uncertainty about disrupting the very thing that we're trying to observe. And that makes me feel even better about what we're doing. So let's power through. These are diatoms. These are silica organisms. This is probably a one that's dividing with probably about 100 microns in size. I love this one, another diatom. The morphology of these organisms is extremely fascinating, and I just find this one's obviously they grow by splitting in half, and I find that to be just incredible, the different shapes that have evolved over time. A linear phytoplankton, our friend the helical diatom, acantharians. These are pretty amazing. They have a very strong spike-like structure, and they use strontium. How they harvest strontium is, like, to me, just an amazing thing. And they catch things on these little spikes. You can see here, this is 100 microns, so this guy is probably around 0.3 millimeters. And they transport the particles down into the center part here. This is our friend, the copepod, the most numerous. Well, there are two candidates for the most numerous multicellular organism on the planet. This is one. Larvaceans, These are pretty spooky. I sent one out last year for Halloween. These are filter feeders that live in a gelatinous house. Here you're going to see the house. I mean, they actually, my friend Kakani Katiha at Monterey Bay Aquarium has had a wonderful article and got some beautiful visualizations. If you're interested in visualizing, check out her work of these sort of tens of centimeter sized larvaceans that play a huge role in, filtering particles that are descending into the water. And then this is kind of a spooky picture. This is a female of those larvaceans, and she actually keeps the head babies in her head, and she actually reproduces by having her head burst out, and then the little babies are suspended into the water column. So it's a lot of adaptations that are interesting in this ocean system where you're basically weightless. So it actually promotes a huge amount of variety of things. So what about doing something useful? Fadib and I talked a little bit about machine learning. It's currently the vogue, to use another fresh word. And we have a large number of dedicated undergraduates and graduates that have I kind of look at it like the lemmings jumping off the cliff. I don't want anybody to be offended by that comment, but there is a cultural aspect to this because it's new and it's interesting. But what really can it do for us? In the case that we have 2 billion images, we'd love to do automatic taxonomy. And so we chose from our 2 billion images. What you're seeing here is, 1, 2, 3, 4, 5, 6, 7, 8, 9 different species. And these species are actually involved in harmful algal blooms. So the idea was we put our microscope in the water, it's running day and night, we look for the harmful algal blooms, and maybe we get an early warning system. And then the question was, well, how are we doing this now? And how well does the machine learning taxonomy correlate with the established technologies that we're using, where you have a person, they go scoop up water, they filter it, they look at it in a microscope, they know what the volume of water was, they count the guys that they see and measure abundance. And could we calibrate our in situ machine learning underwater microscope so that we could do that automatically? Because the microscope has lots of advantages. I mean, it does it 24/7. And so these are unpublished results. And we do see, in fact, linear correlation for the more abundant species. So this is kind of a complicated slide. I don't mean to confuse people. I'm sure if you spend enough time, you'd be able to figure it out. The lower two graphs are showing basically the number of organisms we counted in the lab as a function of time, basically. And then the vertical axis is showing the number of organisms that were counted by the machine learning algorithm. And we were really happy to see for these more abundant species that we had a nice R-squared. So we think we have a way for at least these two to correlate what we're seeing in the lab, in the field, with what we're seeing in the lab. Unfortunately, there weren't a lot of the other guys, and so the correlations are much worse. But we found this to be optimistic, and this is now being written up and submitted in the next few months, and it provides, I think, an interesting link between what we might do and what we're doing now, which I think is an important thing that the oceanographers always want to know. Well, how does what you're doing now correlate with what I did in the past? So let's talk about maker technology. We have a lot of opportunities. This is a little bit of the talk that I gave last year to Fidel's. This is Pichaya Lervitlai, a wonderful student in my lab, and I had this idea that instead of using acoustics to measure velocity of the water, we could use the suspended particles. And Pichaya jumped on that and built this video velocimeter here, what you're seeing, and these are actually the more traditional technologies for sensing acoustics, using a little bit, these are megahertz beams that are sent out, and you measure the Doppler. There's a little teeny volume here, and you're getting 3 components of Doppler from that, and then you can invert velocity. And here's an image of this. So we're doing a stereo camera. We have 2 mirrors that direct the 2 views onto a single camera lens, and what you're seeing is this disparity between those 2 views. Now, what I find really interesting is looking at this and thinking that we see linear flow across the field of view. That's pretty clear. But then when the current reverses, and this is due mostly to wave action, the period of it is due to the wave action. It's causing the subsurface movement of these particles. But things are getting mixed up, and I find the decorrelation scales to be seriously interesting. And then, of course, to do this right, we want to correlate it with what's happening. Again, a complicated slide. These are the X, Y, and Z component of the acoustic in blue, with our imaging in red, and what you're seeing in the velocity and the X and Y comparison are doing quite well. The system actually blocks the flow along the axis of the cylinder. So there's a little bit of blockage, but for the flow parallel to the face of camera, it's really doing well. A fascinating thing is that if we look at signal to noise of the acoustic versus the imaging velocimeter, when particle counts get low, we actually do a lot better than the acoustics. And because the acoustics relies on particle backscatter, when the particle counts are high, we actually don't do quite as well. And this needs, further analysis, but it's just the sort of the tip of the iceberg on that. So that instrument, I think we could build it for, $800 or $900, and the ADV, acoustic Doppler velocimeter, is on the order of $8,000 to $10,000. So there's at least an order of magnitude of, and now we can think about putting many of these out in coral reefs and kelp forests, maybe putting them on autonomous vehicles to see their speed relative to the water, as long as we can get out of the shear zone that's adjacent to them. So we're pretty excited about that. We've already seen that. So I'm not going to talk about it. Pichai actually built another camera on his own, a bit unknown to me. This is during the pandemic. I was home zooming like this, and he's like, oh yeah, I built this camera system. And so it actually is a Plankton imaging system, and it got published in a nice journal, Methods in Ecology and Evolution. He called it the IPAX system here. And what you're seeing now is it's a single view. And then he had two of these, and he sat them on the seafloor. And there's all these crazy organisms that come out at night because they don't want to be eaten. And he was able to characterize those. And so what I'm saying is, there's a huge amount of room now for characterizing ocean microbiota or mini biota using systems that are a lot less expensive, just because of the hardware that's accessible. So moving along here, this is a project we haven't published anything on. One of the great things about being here at Scripps, and I do love it, is having colleagues that are going all over the world and studying things like coral reef. And Stuart Sandin is one such colleague. They have this 100 Island Challenge where they're studying 100 islands and they're mapping 10 meter by 10 meter areas, hopefully every 3 years, and looking quantitatively at bleaching, and also when the reefs can come back, they get re-established, and there's a whole bunch of interesting science that they can do. So you guys are probably familiar, the computer people are, and the ECEs perhaps are, with this structure from motion, which is a relatively decadal or possibly even a little longer stuff that will allow us to, I'm not going to go into the history, but I do find it fascinating. It started with, like tourists in Rome taking, hundreds of thousands of pictures of the Colosseum, and then combining all of those in order to get the three-dimensional structure. And it allows one to solve for pose as well as location, which is a fascinating thing because that's pretty high dimensional vector that you're estimating. So one of the problems is you need to know the scale. So if you took 1,000 pictures of a little thing and 1,000 pictures of a giant thing, without some inherent scale marker inside of the pictures, you wouldn't know how big the thing was. So they invented this very simple target that you're sitting there, and they've been putting that down, and they use that as a marker. But they often don't know. They would like something that was a bit easier to deal with. So Stuart had this idea that we could actually build a underwater instrument that instead of displaying just a picture that was not time varying, that was time varying. And here you're seeing their study. It's a 10 meter by 10 meter area with a bunch of these targets out. And he asked us, could you have an instrument that sat there and displayed a QR code? And that instrument would measure depth and temperature and anything else that you might be able to stuff into this QR code. So it's a really interesting application of information theory. I mean, the QR codes are really sophisticated in their ability to stuff as much information with error correcting as possible. And even though I did take a little bit of information theory years ago, and I'm certainly fascinated with the life of Claude Shannon, I am not by any means an information theory expert. The other thing is you got to see these things and you have to be able to resolve that QR code. So there's a whole bunch of interesting questions related to that. But we did build this. This is actually just one of those CAD models where you imported stuff. Devin Rattel, my engineer, is an engineer in my group. He's a wonderful mechanical engineer, and then he actually built a bunch of these, and we took them to the field. So here it is, sitting in the field in a recent survey, and we are using machine learning techniques to find them, which is challenging because the morphology and structure and texture is the right word that the computer science imaging people use, varies greatly, and depth, things get less red. There's scattering. So it's a pretty fascinating area to be thinking about these QR codes, and could we train a machine learning algorithm to actually figure out what's there under the perturbations of the ambient optical environment. Okay, moving along to our microscopes. So I don't use the word lucky anymore. I prefer to think fortunate. So we were actually fortunate a number of years ago to obtain funds from the Keck Foundation to build underwater microscopes. I was an electron microscopist, and we started building these microscopes in our lab, and I was like, well, this was really my idea. I said, why don't we build something a diver can take down with them? Even though I don't dive anymore, you can see the benefit of such a thing. And so we took our Keck funding and we built the benthic underwater microscope, referred to as the BUM, which was published in Nature Communications. And if you know British slang, it actually is kind of a funny joke. And there's Andy with the BUM. This is the microscope, and this is a pretty funky monitor. Paul Roberts is still working in this area. There's a new monitor, and it's really cool, and it's a 4K display, and it works underwater. And so Andy and Tali Triblitz, who is a professor at the University of Haifa in Israel. Andy is now working with Georgia Tech as a postdoc or a staff scientist looking under the ice, and it's a precursor for the NASA mission to Europa. So maybe they'll meet, maybe we'll go to Europa someday, and we can meet the wife of Jeff Bezos. You might follow the story of her interest in going to Europa, which I found to be interesting, a bit comical as well. Sorry. Anyway, okay, so Fidel's smiling. So here we have the resolution. We want to measure these zooxanthellae. So a coral is a symbiont with, it's a farmer. It farms little teeny 10 micron guys inside that can take light and nutrients and turn that into sugars that the coral can eat. It's an animal. And so we want to have undisturbed organisms, and we really want to do stuff in situ. So this is now maybe 12-year-old technology. What we decided to do at the time, and could be done a lot cheaper, and Tali Triblitz is building new microscopes that I've seen, and they look really cool, and they will be no doubt as good, if not better, and much cheaper. We have LED illumination and objective lens, and the point I want to make here is that what actually potentiated all of this work was the semiconductor industry, which I actually worked in a little bit in the inspection, worked for a company called KLA Tennecore when I was finished my graduate study, developed these lenses, and it's it's geometric optics. We're not talking about entangled photons or anything, to be able to see little teeny things that were pretty far away. So the circuits have been shrinking, 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 and we want to see the high magnification, and we don't want to get too close because we don't want to bonk into them. So when we looked out at the marketplace, there were a number of manufacturers that were actually making high resolution microscope lenses that had nice working distances. So the working distance means like how far away can you see the thing? And here you're seeing, almost 8 and 7 centimeter distances with relatively high resolution. So all of a sudden, the zooxanthellae, which you're seeing here, are now resolvable using a commercially available microscope lens. And so this is another thing you, of course, need a lot of light when you are seeing little teeny things, because it's really the number of photons per pixel. And so if your pixels are teeny, you better stuff enough photons in there so you get enough reflected light to be able to see the thing. And then on the left is an in situ image, and on the right was an image that Tali and Andy put together in the lab. And what's really cool about this, it's a little hard to see. You can really resolve the zooxanthellae and this green tips. So this is a coral polyps. These things are like little tentacles that stick out into the water, and they're almost like little fingers that circulate things. And then these are the mouths of the coral polyp, which allows them to eat plankton if they're not able to get enough photosynthetically derived higher sugars. And we also did some simple stuff which allowed us to do focal stacks and extracting. You can, go into the standard image processing programs and find those kinds of things. And so here's this nice picture that we had. And so we actually sent Andy to Israel to work with my very closest friend, Amatsia Ganin, who is perhaps watching this from afar. And Amatsia was interested in predation. And so one of the interesting things in ecology is the flow of mass and energy through ecological systems. This is the fundamental aspect of ecology. And Matthias was like, well, let's see if they're eating the plankton, the little copepods and other things. And they sort of weren't that hungry, or I don't know what was going on. Could make a few jokes. But nevertheless, then one of the people at the labs are like, well, why don't we put two together that are competitors? So every time I fly into New York City, I see this huge, skyscrapers. And what it's about, we're competing for light and space. And it's the same thing in the sense of the corals. So we put these guys together, and here you're seeing them. What you're seeing is this is not in real time. It's 2.5 hours replayed in 30 seconds. It's a rectangular field of view, but the microscope itself was 2.8 millimeters by so it's 2.8 millimeters by 2.4 millimeters. So what you're seeing is that the coral on the left is, it's basically sort of its intestine. It's a mesenteric filament that secretes proteases. And basically what it's doing is it's dissolving the guy on the right. So imagine you go over your friend's house for dinner and they decide to engulf you and reduce you to amino acids or something like that. It's certainly an unpleasant experience. It's life and death. And if you want a new plot in New York City, you better make sure the old guys aren't there anymore. So we had a lot of fun with this. And I just want to also make the point that when we got this data and it was published in Nature Comms, and I thought, this is really cool. I think the public would enjoy seeing this. So we actually wrote to the New York Times. I'm a New Yorker. I love the New York Times. And we're like, you guys might be interested. And they're like, oh yeah, sure, send us something. So we sent them this, like, wow, this is really cool. So I don't have connections there. What the point I want to make is that if you come up with interesting and cool stuff, you can garner publicity based on the intrinsic interest of the media, which you may disagree with, of course. But it wasn't like I had a connection with people in the New York Times, and I think that's encouraging. So they actually ran a piece that was on their front page based on subscription. They see who's clicking on what. And it was a lot of fun. We wound up, I'm a member of the Acoustic Society, and that's part of the American Physical Society, and it was on the front page of Physics Today. And the Keck people even put it on the front page of their annual report, which made my development people fall in love with me forever. So what's happening now? Well, we're still doing it, even though I'm emeritus. Let's move from anatomy to physiology. Let's look at the transport of mass and energy. And can we do that with a microscope? So there's the technology called pulse amplitude modulated imaging, or PAM. And I can't go through all the details, but basically what you do is you saturate the photosystem, and then you see what it takes to do that using double pulses. And this has been done in the lab. This is being done in the field. So we actually got this idea to do it underwater. And we were fortunate, not lucky, to get it funded. And here's a prototype. I probably could have gone downstairs and photographed and it's the Bunsick underwater microscope with PAM. So I've nicknamed it the Bump. And so this is now working. It's sitting in my lab. We're recruiting a postdoc to work on it. You know, coral reef ecologist we have who's very interested. And it's really moving from anatomy to physiology, and I'm super excited about being able to do that, because that's part of what we want to do, is to look at the transport of mass and energy through ecosystems. Okay, doctor now, Camille Pagniello, who graduated last month, is a participant in this Zoom meeting, and she joined us. Geez, I'm really bad at like how many years ago, but enough years ago to work on a project where we actually deployed a number of underwater microphones, which are called hydrophones. You're probably aware of that anyway, to look at species diversity and abundance using passive acoustic monitoring. And I just love this idea that we can listen and sense what's happening in the ecosystem. And also the sort of idea of being a passive audio hearer, or however you want to call that. You know, many of us have snorkeled and we hear the, you know, there are these fish that sort of crunch on the corals. But there's a lot going on with audio technologies, because sound propagates well underwater, and there's a whole lot of stuff we can talk about related to that. Why two blue whales can say hi to each other across the Atlantic Ocean takes 20 minutes, but hi, 20 minutes later, hi, 40 minutes later. Okay, so Camille was recruited. She did a wonderful job. I better say that, she's on the call, but she really did. And we invented, or she invented, we all invented, a underwater camera system. We took a Sony Alpha 7S for sensitive, and she figured out how to stick it in a housing. And it wasn't so hard to stick it in a housing, but it was more complicated to stick it in a housing and get it to work for two weeks underwater. And she figured all of that out, and she's an excellent diver and really organized person. So she built a system that included the passive listening and also passive imaging. So we don't have to blind the fish in order to see them. We're just, you know, watching them using ambient light. And here you're seeing the result of a 13-day deployment. We collected 17,000 images, and the quantification of that is, did they all have fish? Probably not. Did we? So we actually had to go through them manually to figure out where the fish were. Of course, that presents wonderful opportunities for using machine learning technologies for identifying where the fish are, and even figuring out what fish they were. There are other people, my very close friend Dave Kriegman has worked on a thing called CoralNet, where he's actually looking at using machine learning technologies for quantifying. And there was a conference last week of the ICCV, if you're familiar with that, and there was a session dedicated to underwater imaging. So Jill did a wonderful job. And the cool thing, I don't have the data here, we could see them an hour before sunrise and an hour after sunset. And a lot of action happens when the lights are low, because that's when the guys that are hungry are coming out, because they've been hungry all day, and where the predators are coming out. So it's called crepuscular time. I love that word. And it's just fascinating to me that we can listen and see using passive technologies, and enhanced by this sort of maker technology that we've shown you. And Camille did a really interesting thing. I don't have a lot of time. If you're a signal processor, I will tell you what she did essentially was to measure the impulse response of the medium as recorded by the 4 hydrophones, which had, 6 pairs of time delays, and 4 times 3 divided by 2. And that's a 6-dimensional vector. And she and a wonderful postdoc, Jack Butler, made a synthetic fish. I mean, it didn't look like a fish, but it sounded like a fish. And they knew where it was, and they were able to characterize the impulse response of the medium by knowing where it was as a function of time. And it turned out that really needed to be done because the propagation of sound in this kelp forest is really a bit insane. There's a lot of shading, there's interaction with the seafloor. And so she was able to reduce the errors in what she did. So she's getting closer to a meter, which is really a wonderful way of tracking fish. So can we use species diversity and abundance to with passive acoustic monitoring? And we need the imaging to be able to label. We haven't yet been lucky enough to hear and see a fish at the same time. But hopefully with more time and money, we'll be able to do that. Because many of these, we don't know who's talking. And the answer is yes. We can use hardware algorithms to develop catalogs of sound associations. We hopefully will, there's some of them we know which they are, to obtain knowledge of which species produces which sound, and multi-element arrays to, help figure out the tracking. And of course, time and money, that's your bandwidth. But the interesting thing she found out, even though she tracked only a couple of fish, now one of them was just like hanging out outside of the marine protected area. So if you're going to build a marine protected area, you better make sure that your fish are inside of it. And so we think that this kind of technology will actually be important in helping that. Okay, so last but not least, let's talk about underwater vehicles. It's 12:40. Let's see how much time I have. 12:47. We started at 5 after, so that's 42 minutes. I have about 3 or 4 minutes. I'll try to stay on schedule here. This is sort of a crazy story. I mean, many, many years ago, I was, so Walter Monk, who lived to be, 102 years old, died 2 years ago, was a luminary in oceanography, and basically said, the 20th century is going to be known as the century of undersampling. So that's because we're the outside in instead of the inside out. We got to be someplace. I guess this is on a timer now, so I better be quick. So I was recruited by my friend Simon Levin in 2001, who's a really famous mathematicologist at Princeton, and Jane Lubchenco. And they were trying to figure out where to put these marine protected areas. And can we track where baby fish are born and how they wind up to a place where they can settle? So they like to settle in, areas where there's food, and they prefer certain kinds of habitats. And then being here at Scripps, I was joined by my good friend Peter Franks, who was actually a Woods Hole graduate and joined us. He was actually chair of our department and is now recovered completely, I hope. And he was like, can we measure a swarm of plankton trajectories? So the idea is the plankton get pushed around. That's what plankton means. And Simon and Jane were like, well, can you figure out, passively how little baby fish who can't really swim much wind up in places where they can grow and proliferate? So I decided to invent this autonomous explorer. We had done some previous Lagrangian vehicles, which were much larger, but it seemed like a teeny thing, 1.5 liters. Maybe that's not teeny, but small, using my wife's gracious hands. And we basically deployed a swarm of these. We just had them passive. So we had a bunch of GPS localized transmitters, and they listened, and they were able to use time delay between the transmitted pulses. And that was advocated by my friend Kurt Schergis here as a way of, we just got a proposal turned down. Perhaps some of you reviewed it. We want to build an underwater modem network. We can do transponding now, and we can actually have a swarm of underwater vehicles that go outside of where the GPS transmitters they can be heard. And so they can self-localize and then tie themselves to the rest of the world. So this is Jessica Garwood. She graduated last December-ish, and she, in another experiment that was funded by NSF, the first one was funded by NSF, she actually deployed a bunch of these in the surf zone. And what was really cool was she was able to measure, they were kind of surfing on an internal wave. So if you know about internal waves, there are density fluctuations in the water, and you can see those accelerating. So when she gave her PhD address, basically, and thank you, that's me. When she gave her PhD address, the crowd went whoa when they saw all of those surfing AUEs, and I've been known to dress up like a plankton. So far, it's not crossing any, of the what is it, the nine, the whatever it is, the nine conduct that we're all concerned about these days. So that's it. I'm ready for questions. Sorry if I went a little over.
Wonderful, Jules. Thank you very much. Very exciting work. We have some time for questions from the crowd. I'm also going to allow people to unmute themselves and also to start their videos if they want, changing the sort of the rules. So if anyone wants to ask questions, you can also use the chat and I can ask that question. Alternatively, I'd be able to, you can ask it yourself. I will start with a question. So fascinating work, captivating, actually, I think is a better word to use in this case. One question that I had is in the underwater imaging systems, how long do they last in general? Like I know there are batteries in them, and if you could, and how often do they take images? Do they take continuous images in general?
So are you talking about the one that Camille deployed in the kelp forest?
So
Or
I saw that that one lasts for like 13 days, but in general, sort of what is the limitation on how long they can last? And if you could make them last for longer, what would you want to see, whether it's at macro scale or micro scale?
Yeah, so that's a great question. The thing I like about oceanography is that they're like super fundamental, simple things that you have to deal with, like making a housing that doesn't implode. But the thing with underwater imaging that we find with our peer microscopes is they get biofouled. And the biofouling is a function of the environment that varies quite severely. So we had a big phytoplankton bloom last April, and it was actually a harmful algal bloom because the waters went a bit anoxic. And within 2 or 3 days, I mean, the lenses were just covered. And there's an article in September, October, Photonics journal. I called them schmutz. The lenses get covered with schmutz. And so, on the other hand, if you're going deep in the deeper ocean, and you take a few pictures up on top, and then you go down to 1,000 meters, those can last a really, really long time because the amount of biofouling is much, much smaller. Sure, we would love. I mean, the Heidi Sosik, one of my colleagues and an amazing researcher at Woods Hole, has developed this, IFCB imaging flow cytobot that's become the standard for looking for harmful algal blooms. And to her credit, she developed a whole technology to actually remediate the biofouling while they were pumping seawater through these tubes. And I think the hardest problem she had to solve was how to keep the system from biofouling, and it works extremely well. So I hope that's partly the answer. Environmentally dependent, what would we like to see? I mean, marine snow. We'd love to be able to migrate with zooplankton, which measure their gut contents. Who's eating what, and how long is it taking them? Okay. Let's move on to the next question. So, Wayne Pavalko sent a JPEG of Example from North Atlantic after a few months. So I guess that's a camera that you built, and you're, Wayne, do you want to come on board and tell us the picture? I mean, happy to have you share.
Yeah, can you hear me?
Yeah, totally.
Yeah, so these are from some down-looking cameras, so surface down-looking cameras. So basically right at the surface, looking down maybe 4 feet down. And at least in the North Atlantic, they usually stay pretty clear for about 3 months. And then
Wow.
right around that 3-month time frame, there's both kind of this kind of slime covering, and then also you get these the biofouling that you see there. And those are I forget the exact species, but those are pretty typical, at least for the North Atlantic, to cover the bottom of this particular case that we use for this particular application.
Excellent.
But we can get about 3 months, and occasionally they get cleaned like randomly, that they'll be clean for a while and then they'll get back to being biofouled again.
Perfect.
And is it continuously imaging, or like one image per day? Like how many images does it take?
If you're talking to me,
Yeah.
infrequently, because these come over Iridium. So Iridium short burst data, so they're a little expensive. That image right there would probably cost about $15 to send. But if you want an image from anywhere in the world, you can always use Iridium SVD and just break up the image like we do. So maybe once a week for this particular application. And then we have ones topside too. So just topside cameras, which of course don't foul, but they're used for other applications where you just want to see what the ocean surface is like topside.
So maybe you can post the link and we can move on to the next question. Thank you so much.
Great. Yeah, one other thing that I will share is Dr. Jaffe will also be giving a tutorial on underwater imaging methods starting a little bit after 4. So for anyone who would like to also listen to the tutorial, you're welcome to join us. So we have some time for a couple of other questions, and if people don't ask, I have a gazillion. So I will keep asking.
A Googleplex of questions.
So I actually, in terms of the miniature robots, can you tell us a little bit more about like their design? Because we didn't have as much time. Like, what do they do? How do they move up and down? Do they move around? Like, how are they propelled?
Okay.
Yeah.
So the primary thing was to make something that was so small enough that the Reynolds number, which is the ratio of the sort of inertial component, to make it increase the surface area to volume ratio, so that they were able to track the flow. So they're actually like a pill capsule that can expand and contract to change their buoyancy. And then, so they have no motor for propelling it. So to get them to sink, we just have them shrink. And then we have another piston. We have a piston that's on the bottom. And in this case, nature was very kind to us, because when we want them to come to the surface, we're, fairly, it's fairly shallow, and we can expand them quite large to get them to stick the antenna up so that we can get them back. We get a GPS signal and it tells us where it is. And then when we shrink them and they go down, we can use the smaller surface area piston. And it's fortunate that small changes in density actually modulate, or mediate, I should say, reasonable changes in depth. So their main trick is to basically go up and down. Now, that being said, there was a very interesting control theory problem where we wanted them to hold depth. And we were told by our mechanical engineers, oh, just use a PID algorithm, that works for everything. And it didn't work in this case. So we were lucky to import some brilliant French students from the École Polytechnique, who probably wanted to spend 2 or 3 months surfing here in San Diego. And they were actually able to come up with a 2-state estimator. So the first PID gets it down to depth. And then, since a teeny bit compressible, there's another set of PID controllers that actually allow it to sit at that isobath. And so they are passive, they only listen. They are reasonably well synchronized over a short deployment of a few days, so that we can measure the time delay between, we have 5 surface floats that are GPS localized. We have microsecond accuracy in when those pulses are going out. And it took a lot of work to be able to figure out the time delay estimates for inter-time delay estimates for relative positioning, but it wound up to be about a meter. My colleague Peter has lots of differential equations that he uses. I'm not scared of them, but it's not the way the world I work in, to describe internal wave propagation and pushing that the plankton would come together in the trough or peak of an internal wave and then move apart. And that's what we saw. We actually saw these vehicles pulsating with time delays that were consistent with what we thought were internal waves. And there's a group actually at University of Washington that did something. They were interested in measuring noise levels from their hydrokinetic plants. And of course, when we measure stuff, we don't want to disturb the organisms that were around it. And even though Paul Roberts pleaded with me to use the AUEs in the hydrokinetic, I was like, you're leaving to go to Monterey. Who's going to direct this project? So they built themselves a wonderful set of smaller underwater vehicles that are even less expensive. And I'd be happy to refer you to that group as well. I see ourselves as, I don't want to corner the market. I just want to spark some innovation, basically. Anyone else? Questions or Fadib or whatever you guys are up for? Okay, Brian Lee. Okay, there was a question on long-term monitoring from Syed. And the answer is, I think we were encouraged by Wayne, who might be a good connection. And again, yeah, I think so, as long as the biofouling. So to remediate the biofouling, we've been using a wiper. We have a little bit of copper around it. That's a whole interesting area. I mean, I spent a few years trying to figure out why fish eyes don't biofoul. And I finally found a guy who could tell me why. Because one, they secrete a antibiotic and they exfoliate. So maybe we need, not lenses, but the ports that actually exfoliate. And then, so the biofouling, I think, is an interesting thing. Brian Lee, the suit sensor package. What's the advantages of using QR codes versus how are the QR codes collected? Okay, great question, Brian. So we're sort of fascinated with the technology. I'm not entirely convinced that we couldn't use something simple like barcoding. But the QR codes seem to have a lot of the error correcting stuff built into them. And so that might be a good thing. And then how are they collected? Well, this is the great question, and I'm sorry I didn't make that clear. So Stu Sandin goes out and he collects a bazillion images. And then if he has other data streams, like temperature and depth and current, he's got to then synchronize and combine these. So from the point of view of the person who's doing these coral reef studies, if that information is embedded in the picture, he doesn't have to worry about synchronization. He's seeing exactly what the environment is doing at the time. Okay, so Andre, Rico, do free flow systems have limitations in terms of the size and particles? I don't think so. I can certainly pontificate on that in a speculative way. I think I've been fascinated. I mean, as a physicist, you think of the state variable of the system. So if you have an article that's being, sending photons at a thing, the state variable is sort of all of the information that you can collect, the omnidirectional scatter from that, which could be modulated by, fluorescence or other types of re-emission of. So I think the limitation really is coming up with a way to sense that information. And when I invented that laser sheet imaging system, which ultimately I didn't, I got a little bit of credit when the Nobel Prize was awarded to people that built these laser sheet microscopes. And so that's an interesting question. Brian wants to volunteer, send me an email, jjaffe@ucsd.edu. and Wayne has some other stuff. Wayne, I think if you posted a link to a website, or even give folks your email on the chat, then people who are interested would be able to get in touch with you and learn about the great things that you're doing. I'm fine staying on as long as Fidel is willing to adjudicate my pontificating, and I wanted to thank everybody for coming. And if you have any interest or questions, don't be shy. I'm a friendly guy, always happy, and I'm so sad that I couldn't be there, because that would have been even more fun to spend the day and learn about what you guys are up to, and maybe how we could collaborate on stuff. So
Hopefully very soon. So
yeah.
thanks very much, Jules.